Hi everyone, and welcome back to another episode of Science. In this episode, I want to teach you about how you inherited the traits and characteristics that you did, and how you might pass on these traits and characteristics to your children. It starts with this guy, Gregor Mendel. Now, a long time ago, when he lived in 1822 to 1884, he conducted a series of experiments in 1856 in Austria on pea plants. He was also curious as to how inheritance worked, what were the mechanics behind it, and could he do anything to manipulate or control it. And so he carried out some experiments on pea plants, combining different traits and characteristics together and seeing what offspring were produced. And through the results of these experiments, he was able to construct some theories about how inheritance of certain characteristics actually works. Now, in modern day, we have technology to help us answer these questions, but they have very much proven him to be correct in the calculation of how the inheritance of traits works. If you're a little bit curious as to how he was able to do this so long ago, click on this video and have a little watch of the history of the experiments on the pea plants. But we'll keep moving forward. On this slide, I've got a human karyotype, and if you're unfamiliar with what a human karyotype is, I'd recommend going back and having a watch of an earlier episode. But to very quickly summarize, a human karyotype is all the chromosomes that are found in each nucleus of each cell. And a chromosome is a chunk of DNA. They come in 22 homologous pairs, plus your sex chromosomes. And this particular individual has an X and a Y, making them a male. One chromosome comes from dad, and one chromosome comes from mum. And that's the same for each pair of chromosomes. You inherit one set from dad and one set from mum. Again, if you're not too sure about what I mean by a homologous pair of chromosomes or what a human karyotype is, I'd recommend going back to an early episode and catching yourself up. Okay, let's begin. Here, I've got a pair of homologous chromosomes. Now, for a homologous chromosome, I've got the same genes, the same instructions found at the same location on the homologous pair of chromosomes. However, these genes, although they code for the same thing, you can get different versions of them, alternative forms of the same gene. And we call these alternative forms alleles. In this example here, I'm using the color for flowers. So this flower has a homologous pair of chromosomes, and this gene here tells the flower to produce a certain color. One chromosome has an instruction, an allele, that says make purple flowers. The other chromosome also has instructions for flower color, but this gene has a different version of it, and it tells the flower to make white color, white flowers. They're the same gene telling the flower or telling the plant to produce a color flower, but you can get different versions of it, meaning that one allele, one version of the gene says make purple flowers, and one version says make white flowers. And these different versions of the same gene are referred to as alleles. Sometimes one gene or one allele is dominant over the other, stronger than the other. And so if we were to assume that the purple color is dominant over the white color, then this flower would be purple in color. Or if the white was a more dominant allele, the flower could be white in color. And you can also end up with combinations in between as well, but we'll leave that just for now. Now, what the flower actually looks like on the outside, what it shows to the world, is referred to its phenotype. So you may have blue eyes and blonde hair. That would be your phenotype. That's what you look like. Or you may have brown eyes and brown hair. Again, that's what you look like. That would be your phenotype. Or you could look anything in between, and that would be your phenotype. That's what you look like. Let's have a look at this example down the bottom here. I've got another pair of homologous chromosomes, and this time I've got 3 genes, 3 sets of instructions, telling the individual to create or do 3 different things. You'll notice that we can say this chromosome comes from mum, and this chromosome comes from dad. I'll just put a D here for dad, and a mum, an M for a mum here. They are a homologous pair of chromosomes, meaning that they are matching. They have the same genes at the same locations. But sometimes you can get different forms of the same gene, different versions of the same gene. In this first example here, I've got 2 capital P's. And because they are a capital letter, I would refer to them as a dominant allele. And because I have 2 of the same alleles, I would refer to them as homozygous, meaning they are the same. And so when I look at both of these alleles together, I can see that the genotype for this particular trait is homozygous dominant, meaning 2 of the same big letters, homozygous dominant. Whereas for this particular trait, I have 2 lowercase letters, meaning that this particular trait is homozygous, meaning the same 2 letters, but this time recessive. So I would say homozygous recessive, because you've got 2 lowercase a's. For this particular trait, I've got 2 different alleles. Here I've got 2 different versions of the same genes. I've got 2 different alleles. In this case, because they are different, I would refer to them as heterozygous. They are different. Now, in this case, because I have 1 dominant and 1 recessive, often the dominant allele will overpower the recessive allele. So whatever characteristic this dominant allele represents would overpower this lowercase letter. So for example, we'll go with eye color. So let's pretend a capital B, this dominant allele, codes for brown eye color. And this lowercase b codes for blue eye color. But because this uppercase B is dominant, it will overpower the recessive allele. This means that if someone was born with this genotype of heterozygous, big B, little b, the phenotype would be brown eyes, because the brown eye allele, the capital B, would overpower the recessive allele, the lowercase b. Now, there are many traits and characteristics that are controlled by more than just one allele, but just as an easy example, I can use eye color in this moment here. Let's have a look at how you actually pass on these alleles to the next generation. So in this slide here, I've got a father and a mother. The father has inherited his own set of chromosomes from his parents. So this blue one might have come from his dad, and this red one could have come from his mother. So for this particular trait, you've got a homologous pair of chromosomes, and each chromosome would have a different allele on them. One allele is dominant and one allele is recessive. Same for the mother. The mother would have inherited one allele from her father and one allele from her mother. Both of them have ended up with a heterozygous genotype, meaning 2 different alleles on the matching homologous pair of chromosomes. If you were to do a cross of these two individuals, the mother and the father, combining these two genotypes together, there's 4 possible options you could end up with. There is a 25% chance you would end up with a homozygous dominant child. There's a 50% chance you'll end up with a heterozygous child. And there's a 25% chance you'll end up with a homozygous recessive child. Now, in the case of a recessive condition, a recessive disease, in this example, these parents have a 25% chance of producing a child that shows a disease as their phenotype. They have a disease and they show it. There's a 25% chance they'll produce a child that does not have the disease and does not carry it either. But there's a 50% chance they'll produce a child that is safe from the disease. So in this case, the dominant R means that they don't have a disease, whereas the lowercase r means you have the disease. So the homozygous recessive child, because he has homozygous recessive, because he's got 2 lowercase r's, this child would show the disease phenotypically. They show it to the world. Whereas these 2 children here, they are safe because the dominant allele, the safe allele, overpowers the disease allele. But they're carriers. They're carriers like their mother and their father. So both their mother and the father are unaffected, but they're carriers. They have the dominant allele that overpowers the disease allele, but they're still carrying it within their chromosomes. It's hidden. And there's a 50% chance that they'll produce children, 50% of their children, will be carriers for the disease as well. They'll be safe from the disease themselves, but they could go on to pass it on to their children. And they'll have a 25% chance of producing a child that has the disease phenotypically, which shows it to the world. And 25% chance the child will be safe. Now, most diseases are recessive in nature because you don't know you're carrying this within your DNA. All of us are probably carrying some form of recessive allele in our DNA that codes for a disease, but we don't know if we're passing it on to our children. If you have a family history for a particular condition, it could be worth getting some genetic testing done to find out what your genotype is and what your chances are of passing your disease, hidden or not, to the next generation. It doesn't necessarily have to be a disease though. I just use the example of a disease because it's most relevant to you when you're starting your own family. An example of some dominant and recessive traits, freckles for example, are dominant characteristics. A widow's peak, meaning a V-shaped hairline, is dominant. And a free earlobe is a dominant trait. Have a look at your own ear, or have a feel of your own earlobe. Do you have a free earlobe, or do you have an attached earlobe? If you have an attached earlobe, it's actually a recessive trait. It means that you could have 2 lowercase r's, and in this case, you could have a capital R and a lowercase r, or you could have a capital R and a capital R. In order to end up with a recessive trait, you must have homozygous recessive. You must have 2 lowercase letters. But in the case of a dominant trait, you could end up with 2 capital R's, homozygous dominant, or even if you just had 1 dominant trait, it's enough to overpower the recessive allele. If you've got a straight hairline, that's a recessive trait, and if you've got no freckles, that's a recessive trait. So you would have 2 lowercase letters if you don't have recessive if you don't have freckles. I want to help us start to cross over alleles and characteristics together. Now this is what's called a Punnett Square, and I'll give you some practice questions on Punnett Squares in class. But let's quickly run through it here and now. For my parents, one parent is able to hear and the other parent is also able to hear. So the ears are working. However, they are heterozygous, meaning that they have a dominant allele and a recessive allele. Dominant and recessive. The lowercase allele, the recessive allele, means deafness. Deafness. But the uppercase allele means that you can hear just fine. So both of these parents can hear just fine, but they are also carrying the recessive hidden deafness allele. Now each of these alleles, each of these chromosomes, each of these alleles found on a chromosome will be passed into either a sperm or an egg. So this allele could come into this sperm here, and this chromosome with a recessive allele could come into this sperm here. And for the mother, this chromosome with a dominant allele could come into this egg. And this recessive chromosome with the recessive allele could come into this egg. And once you've figured out which allele can go into which gamete, sex cell, sperm or egg, you can do this little cross, a Punnett square, to figure out what percentage your children will have in being deaf or not. So in this case, when both of my parents are heterozygous, I produce a sperm with an allele that has a dominant allele, a sperm with a recessive allele, an egg with a dominant allele, and an egg with a recessive allele. If I was to cross them over and combine them, so this sperm fertilizes this egg, I'd end up with a 25% chance of having a homozygous dominant child for hearing. So this child can hear and does not carry the deafness allele. So this is the ideal outcome. There's a 50% chance though when a recessive allele fertilizes with a dominant allele, a recessive sperm with a dominant egg, they will end up with a heterozygous child, meaning they'll be able to hear, but they carry the allele for deafness. They are a carrier. When you cross a dominant allele sperm with a recessive egg, once again you produce a carrier, a heterozygous child. If unfortunately, however, you end up with a recessive sperm with a recessive egg, end up producing a deaf child where they are homozygous recessive, and so there's a 25% chance of that happening. We are going to practice these in class, so if you haven't received the practice questions from me, please do come and grab them from me. Now, as I said before, there are many characteristics that are controlled by many genes and alleles. We're not going to go into this level of detail, but as an example, skin color. There's a whole wide spectrum of skin colors and tones, and that's controlled by many factors. For Year 10, we're just looking at monohybrid crosses. So we're just looking at one trait, one gene, just hearing with two alleles. But just know that it can get more complicated. So this would be a trihybrid cross. We are just doing a monohybrid cross in Year 10. So this one. Let's have a look at some examples of conditions. So recessive disorders. Most human genetic disorders are recessive. Why? Because the fathers and the mothers don't know they carry them. They have children without knowing they carry the allele that codes for a disease. And so they pass it on to 75% of their children. 50% of them are carriers and 25% of them actually show the disease phenotypically. And then because this person is recessive, we'll use two lowercase r's, you're guaranteed, if this person has a child, they're guaranteed to pass on a recessive disease allele to the next generation, no matter what the other parent is. Even if the other parent ends up being two capital R's, homozygous dominant, their children are going to be 100% carriers. All of their children will end up with at least one lowercase r. And so it gets promoted through the generations. Also, no, we can stop there actually. I was going to do a Punnett Square, but it's just a bit difficult with a mouse. Again, if you haven't received the Punnett Square practice questions from me, please do come and collect that. I also want to show you a dominant disorder. Now for dominant disorders, only some of them are dominant. And that's because if you are born with a dominant allele disorder, you'll show it from the start. You'll probably show it from birth or from early age. So you'll be able to make an informed decision before starting a family, or potentially you won't be able to start a family. And so dominant disorders are not quite as common as recessive disorders. Recessive disorders can hide and they can skip generations, whereas dominant disorders can't really do that. You show them from the start. An exception to this, though, is Huntington's disease. Huntington's disease doesn't begin until middle age. So even if you are born with this, you don't know it until after you've had children. And it's especially problematic because it's a dominant allele, it overpowers the recessive alleles. So in this example, I have a heterozygous parent with the Huntington's disease here, the dominant allele. Now the other parent is homozygous recessive, which is good, which is safe, that's what you want. But if they were to have children, this time 50% of your children will have Huntington's disease and 50% will be normal. If this parent was homozygous dominant, not only will they have Huntington's disease, even if they had children with a homozygous recessive parent, 100% of their children, when you do the Punnett Square, will have Huntington's disease. And they won't know it until middle age because it doesn't begin until middle age. So again, if you have a family history of any of these serious conditions, it's worth considering getting a genetic test done before starting a family of your own. I've got a case example here, a case study example for you here as well. If you're curious about it, I'd recommend you click on this link here and have a look at this news example of how genetic testing can be used when you're preparing to start your own family. I think I'll wrap it up here. If you haven't already, please collect the Punnett Square Goodness booklet for me, where it's got the practice questions. You can go on and take some notes from Jacaranda. These are the pages, and you can also do some practice questions on Education Perfect. Thank you for watching this episode of Science, and I will see you next time in class.